The discoveries of Dr. C. Ronald Kahn have been central to forming our understanding of the mechanisms of diabetes. At the 2013 Joslin International Symposium on Diabetes, researchers from around the world have traveled to Boston to talk about their own research and to honor the man who has made such tremendous contributions to the fight against diabetes. We spoke to Dr. Kahn about his career, his discoveries, and what he sees as the future of diabetes research.
So with respect to areas that I've worked in that I feel I've been able to make an impact on, it starts really at those NIH early days. At that time, we really knew that insulin was an important hormone regulating metabolism, but believe it or not, nobody had any clue as to how insulin worked. It wasn't even known whether insulin had to enter the cell to work, that it worked on the surface of the cell, whether a fragment of insulin was released or bound to the cell. And what the team at NIH, led by Jesse Roth and myself and others, did is we actually discovered the insulin receptor. That is the protein on the surface of the cell that recognizes specifically the insulin molecule. And that recognition allows insulin to trigger and alter the metabolism of the cell. So this is the first step in insulin action. Right before I left NIH, and as I was coming to move to the Boston area, I think was my second important discovery, and that was that the insulin receptor signals the cell through what is known as a tyrosine kinase activity. This is a technical term meaning that the insulin receptor is an enzyme, and when insulin binds to it, this enzyme gets turned on and stimulates a chemical reaction in the cell that chemically modifies intracellular proteins that are called insulin receptor substrates. And we were able to first discover, we were the first lab to discover the insulin receptor was a tyrosine kinase, and with the fellows that were here in the early days of my stay at Joslin, we also described the first insulin receptor substrate, actually both of the first insulin receptor substrates, and now we know there may be many other substrates, but the two major substrates were the ones we found. We also were able to show how when these substrates get modified by the insulin receptor, this is called phosphorylation, this can lead to signaling inside the cell. Now, why is all this important? Well, it's important because, first of all, it tells us how does insulin acting on the cell stimulate glucose uptake or glucose metabolism or lipid synthesis, protein synthesis. It also gives us the first step to understanding what goes wrong in diabetes. In type 2 diabetes in particular, the primary problem is insulin resistance. That is, the body makes insulin, but the tissues of the body don't respond normally to it. And of course, you can't understand that process unless you understand how insulin would normally work to begin to understand what goes wrong in type 2 diabetes. And so we were also among the first to really dissect the pathways of altered insulin signaling that occurred in obesity and type 2 diabetes, to begin to understand also that these pathways are abnormal even many years before the type 2 diabetes develops. So that if you look at children of type 2 diabetic parents, you can study them sometimes 15 or 20 years before they develop diabetes, and they already have insulin resistance and some abnormalities in signaling. So this gives us a way to think about how does diabetes progress, and could we even block this progression before the disease develops? It's not only treatment, but prevention that I think is so important. There have been a number of other really exciting developments in the last few years, and I'll just mention two of these that I think have been exciting to me. One is that we now know that insulin acts on every tissue of the body. When I was a medical student or in early days of fellowship, we thought that there were insulin-sensitive tissues and insulin-insensitive tissues. So the muscle, liver, and fat were the main insulin-sensitive tissues, and we used the brain, the kidney, the intestine as examples of insulin-insensitive tissues. Well, it turns out that we were wrong. In fact, all of these tissues respond to insulin. Some are more important for glucose metabolism, but some insulin plays a different role. For example, recently we've shown that insulin plays an important role in the brain in control of neural function and behavioral activities. And in fact, when you knock out the insulin receptor, when you make insulin resistance in the brain, you can make mice develop symptoms of anxiety or depression. They will have behavioral changes. And there's a lot of interest now in how insulin resistance and type 2 diabetes might be linked to Alzheimer's disease and other neurodegenerative diseases. The other thing I would say that's become very exciting is related to obesity. We have a large effort in this area as well. Again, going back when I started working in this field, really what we knew was that mostly there was a problem with obesity driving diabetes. We knew it created insulin resistance, but now we know much more about the biology of fat and how this might play a role in insulin resistance. For example, there's white fat, which stores energy, and brown fat, which burns energy. And we now understand a lot about the signals that make this differential. Even in white fat, there's harmful white fat, intra-abdominal white fat, and then there's white fat that seems to either be neutral or in some cases maybe even good for you, and that's subcutaneous white fat. And our lab has done a lot of work trying to understand what it is between these different kinds of white fat cells that can make such a difference in metabolism.